All right. Hello, everyone. Thank you for joining us. My name is Haley Rivers, and I am the Kupu AmeriCorps member for the HETF, also known as the Hawaii Experimental Tropical Forest. My position is for the year of 2020, and I am the head coordinator for this webinar series. This webinar series exists so that the community can learn about the research that happens within the HETF, and so that our researchers have a chance to give back and share what they are passionate about. Thank you all for tuning into the webinar today. We are immensely grateful for your enthusiasm and support. Also, please note, we have received more interest for this webinar than ever before for our webinar series, so we may have some weird technical glitches, so just bear with us. A few things before we start. This webinar is being recorded and it will be uploaded to an ongoing YouTube playlist. You guys will receive an email after the event to that YouTube playlist and to our webinar for October. Feel free to toggle between gallery view and speaker view during the webinar. Once the webinar begins, you will be muted and your video is disabled. So if you have any questions, please put them in the chat. They will be answered as part of the Q&A at the end of Julia's presentation. During the actual Q&A section, you should be able to unmute and turn on your video to ask any questions if you prefer to actually ask with your face and your voice instead of just over chat. After the webinar, we will be sending out a feedback form, so please take a few minutes to fill it in. These kinds of events are, you know, a coordination of everyone, and we want you guys' feedback so that we can continue to make it the best webinar that it can be. With that, I would like to introduce Julia Ho. Julia is a research technician with the USGS. She is currently focused on the Hawaiian Hoary Bat, and she works on the Big Island. So I hand it over to you, Julia.
All right, thank you, Haley. Thank you, everyone, for joining. This is a big turnout, very exciting to learn a little bit more about our ongoing research and then other work that we've done in the Laupāhoehoe area. Can everyone see my presentation screen? Good to go. Okay, so I work with the USGS, with the U.S. Geological Survey at the Pacific Islands Ecosystem Research Center, and I'm studying the Hawaiian Hoary Bat, Ōpeʻapeʻa, and this talk will focus on the Laupāhoehoe area. Firstly, I'd like to thank all of these members of our bat hui who are contributing to our ongoing research, or have contributed to our ongoing research. Science is never a solo endeavor, and field biology in particular kind of takes a village, so thank you all. Specifically, I'd like to thank Christina Montoya-Iona and Karina Prinzari, who provided the framework for the slides and for much of this presentation, and then them, along with Karen Corto and Marcos Gorison, provided much of appreciated feedback in putting this talk together, so thanks to them. A little outline before I get started. So I'll give a brief introduction to Ōpeʻapeʻa, our bat here in Hawaii, and then I'll talk a little bit about how we research bats here. Then I'll dive in a little bit to the project of our current research. We are currently working on a project on the Big Island, so I'll focus on the fieldwork for that with a Laupāhoehoe focus. And then lastly, I'll discuss some of the publications that are already out there from previous work that's been done, including bats in Laupāhoehoe. So a quick introduction to the Hawaiian Hoary Bat. I'll build a little bit of an info card here so you have it all in one spot. The Hawaiian Hoary Bat is in the family Vespertilionidae. It is in the genus, it was previously in the genus Laziurus. In 2017, Baird and co-authors suggested that our Hawaiian Hoary Bat and several other related bats should be their own genus, Arestes. While this point is still being debated among bat biologists, they did provide some convincing evidence that the Hawaiian hoary bat should likely be its own species. So if that's the case, then the full species name will either be Arestes simotus or Lazarus simotus. Regardless of what comes out of that, here in Hawaii, it will still be known as Ōpeʻapeʻa. This comes from the wing shape, and that naming has been attributed to the half taro leaf or a canoe sail. So in this diagram here, you can see how it kind of looks like the half taro leaf. It is the only native terrestrial land mammal in Hawaii, and it was recognized as the official state land mammal in 2015. It's related to the North American hoary bat. And so how did the North American hoary bat make it all the way to Hawaii? Well, the bat that lives in the continental US has fall migration patterns, and body conditions change for that time of year. So they put on more fat, they get a little heavier, and that fall migration body condition, coupled with the September and October wind speeds and direction, allowed for probably many successful long-distance dispersals to Hawaii, and this was modeled by Bonacorso and McGuire in 2013. So once they got here, on the left-hand side, we see some photos, two photos of the Hawaiian hoary bat, and on the right-hand side, the North American hoary bat. The hoary bat gets its name from the hoary frosting on its fur, so those kind of white tips. Almost looks like a 90s boy band coloration of fur there. And the one on the right shows the sort of the hoarier color of the Hawaiian hoary bat, whereas the one on the red shows a reddish coloration. So we have two different types of fur coloration here, but the cause for this is still unknown. It does not seem to be related to sex or age. It might possibly be related to genetics. Also, after bats arrived, there was a reduction in body mass, so they're about 30% smaller than their continental relatives. And there was also a reduction in forearm length and wingspan. And this is pretty common for mammals that move to islands or that are isolated on islands, that they become a little bit smaller in size. So next up, they are nocturnal. They're also insectivorous, so they only eat insects, primarily flying insects around in the landscape. And they use echolocation to navigate in the night and catch those insects. So they're flying around at night, trying to catch insects. They produce this very loud ultrasonic echolocation call, and they wait for that to bounce back off of various things in the environment. If they're lucky, it bounces back off a moth or a beetle or some sort of insect for them to hone in on and capture. Lastly, our bats here in Hawaii are foliage roosters. So you might have heard of bats roosting in caves or houses or belfries. The ones we have here tuck up in the leaves of trees, and they have a furred tail that they use to sort of wrap around themselves, and they just hang among the leaves. Their reproduction, I'll see if this video will work. It didn't work when we were trying it, but it's a video of a couple of pups, and they're sexually dimorphic, so the females are larger than males, but there is some overlap between the sexes, especially in juveniles. So particularly females can be similar size to male bats. The females have 4 nipples, 2 on each side, which helps to feed those twin pups that they have. And here in Hawaii, as well as federally, they are listed as endangered as of 1970. The primary causes for this listing were an unknown distribution, unknown population size, and then also potential habitat loss. Potential threats, as listed by the U.S. Fish and Wildlife Service, include collision with wind turbines or barbed wire fencing, loss of critical habitat, potentially pesticides, or predation. So a few of the management questions for these bats include how many bats are there? What are their population trends? Are they widely distributed, narrowly distributed? What kind of activity patterns are there and their population size? What are the limiting factors? Is food and shelter limiting? How do we restore habitat for bats? How do we assess bat use? If we do put together restoration locations and in forested areas, how do we determine if that was successful? So now I'll get into a little bit of the research techniques that we, with USGS and then the Hawaii Cooperative Studies Unit, our partners, have used previously and are currently using to research bats in Hawaii. I'll spend a little bit of time describing each technique, and then I'll show how these techniques are contributing to our current research. So first, acoustic monitoring. I mentioned that our bats echolocate to navigate and catch prey. We can eavesdrop on those behaviors to monitor the bats in the landscape. Some of the calls that we've recorded are search calls, so those are longer, louder type of calls that they use to navigate around the landscape. And then once they start to hone in on something like a food resource or a water resource, they use more of these like foraging calls that are pretty rapid, and that allows them to get a faster response from the call, a faster echo that comes back, so they can really hone in on what they're looking for. And then finally, social calls. These are calls that are believed to have some role in communication between bats. We use acoustic monitoring around the landscape and have used it at many different sites, including DoD installations, some of the national parks, and in forest restoration areas. There's a specific microphone that picks up bat calls, and it allows us to determine bat activity, so presence and then patterns of that on the landscape. We've done acoustic surveys with partners on many different Hawaiian islands. This acoustic work, paired with historical observations collected since 2005, increased our knowledge of bat presence, and we now know that they are distributed statewide on all the major Hawaiian islands. Another technique we use are bat captures with mist nets. Here's a few photos of mist nets, and then in the center is a bat that we're untangling from one of the mist nets. We kind of put them up on the landscape and hope that a bat flies in the direction that where we have the nets and gets tangled in the net. Once we've captured the bat, then we can process it for scientific information and prepare it for radio tracking. That includes weighing it, taking wing measurements and photos, biological samples, marking individuals, and attaching radio transmitters. And then once all of that is done, then we release the bat, and it flies off, and we have to try and find it somewhere on the landscape. So the next part is tracking its movements. We use antenna and receivers and driving a lot around the landscape to try and pick up a signal from the bat roosting in a tree during the day or flying around on the landscape at night. If we are lucky enough to find a roost tree, we look for bats in the tree both with thermal and visual confirmation. Sometimes we do emergence at dusk to see if the bat flies out of a tree and determine which tree it was using. We then later come back in and collect more specific roost tree and forest stand metrics. Another way we monitor the bats is with video equipment, thermal, and also like true color video monitoring. And we've done work with this at wind turbines in bat foraging behavior, and then also at roosts looking at emergence and return time, and then night roosting behavior as well. Coupled with our bat work, we look at the insect prey that the bats may be consuming. So we do some insect prey sampling, compare that to the guano contents available in prey. We've also been collecting caterpillars, and we have done this in the past to determine host plant associations. And then from those, we're trying to link sort of the plant that allows the caterpillar to survive, to become a moth, and which of those moths then show up in bat diet. With the guano and comparing it to insect collections as well, we have done some guano dissection in the past and are currently working on some genetic barcoding as well. Some of the types of insects that we've found include moths, beetles, stink bugs, and termites, all in bat guano or bat poop. So our current project that we're working on, Conservation Biology Project, started in 2018 and is running through 2021. It employs many of these techniques across the eastern part of Hawaii Island. We have year-round sampling efforts at 8 sites, which are indicated on the map here with yellow stars. With each site, we sample it 3 times per year, with some additional netting as time permits. Elevation at sites ranges from 67 meters to 1,784 meters. So we're spanning elevation gradients and then also spread out pretty widely on the landscape. Each site is visited 1 to 3 nights per sample, and depending on our bat captures, we net either 1 to 3 nights. We also simultaneously are collecting insects on 2 of those nights to compare to diet samples later. And I'll discuss that in a little bit more detail in a moment. So thus far, with our captures, we have netted 166 net nights and captured 93 bats through August of this year. That total netting effort equates to about 0.03 bats per net hour, so it takes quite a bit of effort to catch the bats here in Hawaii. Our insect light trapping, again, we've done 2 sheets at each of the sites, and we're doing concurrent insect light trapping with netting on 70 nights so far, and we've had concurrent guano samples for 41 light trapping instances. With our capture effort, we've also been able to recapture some bats. These 6 are the ones that we've recaptured thus far. The bottom 3 here were actually captured from Laupāhoehoe in that location. And our recaptures have been separated by as few as 6 days and then as many as 335 days. So over a year later that we recaptured an individual. This map and graphic shows our pie charts of our capture of males and females at the various fixed sites throughout our sampling period thus far. Most of our sites, we've caught more males than females. We even have a site where we've only caught males to this point. If you look in the top portion of that map, that's where Laupāhoehoe is located. And it's the only site where we've caught more females than males throughout this time period. Looking at that a little bit more in detail, so this is the number of bats captured by sex across the year, so January through December. There seems to be sort of we have some months that are more successful than others, and we seem to catch more females in the summertime than in the winter. This is the observable reproductive condition in adult male Hawaiian hoary bats by month. So January through December again. The light blue coloration is non-reproductive males, so not observably in a reproductive state. And then the darker blue is the reproductive males, and we've primarily caught those in the late summer and early fall. And this is consistent with what we see in this group of bats in the continental US as well, is that the males and females reproduce in the fall, late summer, early fall, and then the females store the sperm through the winter and become pregnant in the summertime. So these are our observable reproductive condition in adult females for the time period of this current project. Light blue, again, is non-reproductive, not observably reproductive. Green is pregnant, and then yellow is lactating, and that red one is post-lactating. Some of these non-reproductive ones may be early in the pregnancy state and they're just not observable as pregnant at the point of capture. I do want to highlight that captures at Laupāhoehoe during our current study of efforts as well as previous work have added a lot of information to what we know about the reproductive phenology of Hawaiian hoary bats. So here's a little graphic of just our Laupāhoehoe captures. So as you can see, 2 of our 3 pregnant bats were captured at Laupāhoehoe and 9 of 10 lactating bats. We also captured 3 juvenile bats, which are not included in this graphic, but does speak to the reproductive state of a lot of the individuals at in Laupāhoehoe area. We've collected a lot of samples during this process as well. We take standard metrics of all our bats, which includes the capture time, their mass, forearm length, age, sex, reproductive condition, and then we put bands for individual identifiers on the forearm to be able to identify the bats in the roost after we've released them, and they don't have a transmitter on anymore. We've additionally collected guano, tissue, fur, and wing photos of bats, and we have quite a few samples from different areas across the eastern part of Hawaii Island. In addition to capturing bats for this project, we are interested in their roosting ecology. With that, our objective is to identify and characterize roosts. We're looking for the locations and habitat where roosts are, and then tree attributes, and also assessing fidelity and if bats reuse roosts and how long they seem to use roosts once they're using them. We attempt to spot bats in the roost with binoculars, scope, and thermal imaging cameras. We even sometimes set out video cameras to see if we can spot the bat coming and going from a roost. We classify roost habitat by looking at the canopy and understory tree species, elevation, slope, the tree species of the tree that the bat is roosting in, the tree height of the roost, diameter at breast height, percent canopy cover, and then the roost height and roost aspect. And from these tracking efforts and roost efforts, we have conducted 486 tracking events, so it definitely takes a lot of personnel and manpower to get to this roost information. And this is all as of 2019. We had tracked 38 bats to 52 roosts and 18 bats to 24 roost trees, and so 52 roost stands. So some of the areas we're not able to gain access, or the terrain is too difficult, so we're not able to get to the specific tree, but we do still collect information on the stand that the bat is roosting in. So progress through 2019, these are in blue are approximate roost stands, and then in orange are confirmed roost trees. That patch at the top of your screen is the Laupāhoehoe area. So we've been able to get quite a few bats tracked to stands and trees in that area. This effort has continued through 2020, and we'll continue adding to this data set as we track bats to roost trees. Often bats captured in the HETF are found roosting both within and in surrounding areas. So right here, I want to thank the folks in the Laupāhoehoe area who have offered us access to their private land and allowed us to come on. That's really been beneficial for us being able to find some of these bats roosting in trees, sometimes in people's backyards or fields. So that's been really amazing in the Laupāhoehoe area, particularly, but all over the eastern part of the island. Some of the roost species, tree species that we've seen bats in are listed here. Particularly Laupāhoehoe, we've found bats in ohia trees, ironwood, and eucalyptus. We described a lot of those tree metrics that we look at. So tree height, canopy cover, perch height, perch canopy cover, and then perch aspect. We found that tree height averages 21 meters, and perch height averages 14 meters. And then we've also found that the perch aspect seems to be somewhat southward or southwest. We use a pretty interesting technology called Pictometry, which looks at aerial photographs and allows you to measure heights of trees in a stand without necessarily having to be in the forested area itself. And this has allowed us to create some sort of a picture of the roost stand metrics. So the forest stand canopy height has averaged 24 meters, and then the cover classification as well. We have quite a few closed canopy locations, but then also some scattered and very scattered locations as well, where bats have been found roosting. Our current work is also looking at maternity roosts specifically. We had 3 maternity roosts that we monitored in 2019 reproductive season. One roost had 1 bat and 1 pup. It was in an urban habitat, lychee tree. Another roost had 1 bat with 2 pups, and this was a mixed native and non-native plantation eucalyptus species tree. And then the last one was 1 bat with 1 pup. It was non-native agricultural and plantation habitat, and this bat was roosting in these bats were roosting in a paperbark tree. Again, we've monitored these same 3 maternity roosts from last year, in addition to some other newly discovered maternity roosts. 2 of the 3 roosts monitored in 2019 were used again by a bat and at least 1 pup in 2020. We're currently monitoring those and continuing to monitor those, as well as the other roosts that we've identified as maternity roosts in 2020. So this probably, we'll see if this video works, I'll play it, but there's a light spot of coloration here in the top. This is a thermal video of a mom and pup at a roost. And if the video works, you'll see the mom fly away, and then there's still a smaller thermal spot there, and that is the pup that stays after the mom leaves. So from this thermal video monitoring of maternity roosts, we've determined that the departure time for bats is thus far has been anywhere from 53 minutes before sunset to 20 minutes after sunset. And the mean time for that was 2.7 minutes before sunset. In terms of arrival time, so returning to these maternity roosts, we had arrival time 41 minutes to 11 minutes before sunrise, with a mean of 22 minutes before sunrise. In 2019, we did not observe any predation at maternity roosts. We also, in 2019, collected acoustic recording, and we had some acoustic recordings that were recorded we recorded during the daytime, so sunrise to sunset, and we didn't find any midday acoustic activity recorded at these maternity roosts. During our current project, we're also using some genetic techniques. On a couple of our current projects, we're using genetic techniques to help understand some of the hidden aspects of bat ecology. We're determining some demographics information, specifically with bat carcasses that are submitted. My colleague Karina Panzari worked on establishing a process to determine the sex through genetic techniques. And then we're also looking at population structure and size, and also focus more specifically on our genetic techniques with diet here. We're doing diet metabarcoding of Hawaiian hoary bats. So each of these vials in this photo contains bat guano in the process of extracting DNA. I always find it very interesting, the array of colors that come out in this in the guano samples. So what is metabarcoding? We extract DNA from the environment, so from either the gut or the feces. In this case, we're extracting it from a guano pellet from a bat. And then we use genetic markers to identify insect prey. The main advantages are that you can get a taxonomic species level prey identification of items in the guano. So you can really understand what specific items are in the guano. You can process many samples at the same time. And then there's also a greater ability to identify soft body prey that with the guano dissection may not be intact. Getting reliable results, however, requires the knowledge of the ecological system under study. So what prey are available for the bats to consume. That you also need to know the analytical procedures used in the process, what genetic markers will be used to determine prey items in bat guano, and then whether or not the local insect fauna is genetically characterized so you can properly identify prey. So if not enough of the insects have been genetically sampled, then it's difficult to pull those DNA, that DNA information from the guano sample. Here's an example of one of our bats from our current study. This is a female bat captured in August at a low elevation site near Hilo in a habitat with mixed native and exotic vegetation. So the first pie chart shows the relative prey availability sampled around the capture site. So this was from the light traps on the sheets, what prey were available for bats to eat on the landscape. The second graphic, there's a large amount in this prey availability graphic of Hemiptera and Coleoptera, but then there's also some Lepidoptera and Diptera. The second pie chart illustrates the OTUs of diversity on insects recovered in the guano sample. So how many different types of prey were detected in the guano sample. So here we see that Lepidoptera and Diptera are representing more of a diversity in the diet, but we also see the presence of similar orders as were at the site. But yeah, you can see the proportions are different here, so more of the Diptera and Lepidoptera. The last graphic shows the sequence count looks like for these OTUs. So here we see mostly sequences belonging to Coleoptera. And so this is just one example from one bat. On our current project, we have collected guano from 63 individual bats, 20 females and 43 males. Our first year of guano samples have been analyzed thus far. We also are working on creating a local insect prey barcoding library. So there are bigger barcoding libraries that are available. However, a lot of the insects in Hawaii have not been extracted. The DNA has not been extracted, and they've not been added to those barcode libraries. So we're creating some of those, and then we'll make them available once the project is completed. And then our insect collection efforts, we've thus far sorted the insects that we've collected from those light sheets to order and family, focusing on Lepidoptera, Coleoptera, and Diptera. And we are also looking at prey host plant associations. So we've collected caterpillars at sites and are rearing them on the foliage that they were collected from, and hoping to make those connections between the plant and then the caterpillar, which becomes the moth, which the bat eats, or the beetle, which the bat eats. So Laupāhoehoe bats in publication. This is sort of the last section. We don't have any results from our current work yet to present, but I wanted to show you some of the ways that bats in Laupāhoehoe, potentially in your backyards if you live in that area, have contributed to science and our understanding of the species in general. One study that was conducted in 2008 was looking at the bat detectability and occupancy with multiple automated echolocation detectors. So they sampled locations in Laupāhoehoe and Umikoa. They found variable bat activity, but with repeated nights of sampling, they were able to estimate detection probability and occupancy at those various sites. This research also assessed the effects of sampling effort, so how many sites and how many repeated nights of sampling were needed to get a good picture of that variable bat activity. And this study really led the way to a bigger 5-year study of Hawaiian hoary bats on the island of Hawaii. And you can see the red dots on this map. Those are all of the various sampling locations across the island. Acoustic detectors were set up at these various locations, and they found that Hawaiian hoary bats are widely distributed. They have a fairly high occupancy in all regions, but the occupancy peaked around the 15th of September, which is expected, especially with the August to September fledging season. So that's when the pups are starting to fly, moving around the landscape. And the lowest occupancy was seen in December and March. Laupāhoehoe was included in this study. So this is a graphic of the detection probability across months. And each of those colored lines is a different site, a different location. The black in the center is sort of the average detection probability for all sites across the season. The purple line, however, at the very top of the screen is the acoustic detection probability curve for the site at Laupāhoehoe. Work in Laupāhoehoe also contributed to several graduate students' projects in partnership with USGS. Studies by Chris Todd, this one effects of prey abundance on seasonal movements of the Hawaiian hoary bat, and also Riley Bernard, dietary overlap between the invasive koki and the Hawaiian hoary bat on the island of Hawaii. Both of these students used guano dissection to look at insect prey in relation to seasonal movement, and then also dietary overlap with koki frogs. So here you see some, on the left, a photo of the guano dissection in process. In the center, those are some of the pieces of insect that show up in the guano that then they were able to identify the insect that the bat had eaten from those fragments of insect left. So when the timing for when bats move to Hawaii is still being ironed out. These are some papers that used tissue samples from Laupāhoehoe to kind of assess when bats first arrived. These two studies and subsequent studies will probably help us understand when bats came here and how they spread around the island. And thanks to 10 samples from Laupāhoehoe were used in these studies, and then additional samples are collected right now that will be used in future work. So we believe from this work that bats arrived roughly 1.35 million years ago to Hawaii. They likely landed in Maui and then spread up and down the island chain. There's a belief that there were expansions at a few different time periods in the population. However, the complete history remains unclear. So for future work, we have collected 60+ Laupāhoehoe bat tissue samples that will contribute to answering some of these questions. And the Hawaiian hoary bat as a separate species, so not as a subspecies of the mainland continental bat, has still yet to be described. So the morphology, echolocation calls, and coloration differences that we see here in Hawaii are still in the process of being evaluated. So quickly before I wrap up, I wanted to revisit those management questions from the beginning of the talk and highlight how our research, past and present, is contributing much needed information. So one of those questions was, how many bats are there? What are the trends? Our landscape scale acoustic monitoring has helped to understand distribution, genetic techniques to improve understanding of population dynamics, and then we're also collecting phenology and life history information. So what are the limiting factors? We are quantifying prey availability and diversity across multiple habitat types. We're looking at what roost requirements that bats might be selecting for to understand how to better build a landscape that has those type of roosts. And then we're also doing video monitoring to identify potential predators at roost sites. How do we restore habitats for bats? Again, we're looking at roost, tree, and forest stand selection. We're characterizing maternity roosts and learning about fidelity to various roost types. And then we're helping to better understand prey availability and how that relates to diet, if there are differences across the landscape, elevation, and habitat types, or differences between sexes and reproductive status. We're also identifying host plants of insect prey. And then the last question, how do we assess bat use? Throughout all our work, we are refining and testing out new bat monitoring techniques, looking at the pros and cons of each, and then how best to implement them in Hawaii. So I want to say mahalo nui loa for your time and attention. Thank you so many people for attending. I want to give a special thank you to the people who provided us land access. So there are many private landowners as well as the Hawaii DLNR DOFA section and then the land division as well. The U.S. Forest Service, particularly the Hawaii Experimental Tropical Forest, which has allowed us access into the Laupāhoehoe area. Fish and Wildlife Service for Hakalau access, and then the Hawaii County and Hawaii Department of Homelands. I also want to thank our funding sources and support sources as well. We've received funding from a few different agencies and then support as well from USGS and the Hawaii Cooperative Studies Unit. I want to thank again researchers and technicians and volunteers, past and present, for contributing to our understanding of bats and their Hawaiian habitat and how they use this habitat. And then finally, of course, to the bats, thank you for letting us interrupt your evening flights and follow you around. And with that, our science can be found here and it is publicly available. So if you have any further questions about any of the research I presented that was published, this is where it can be found. Thank you very much.
Do that. Clap again.
Thank you so much for that amazing presentation. So with that, we will begin the Q&A section. I will start us off with a couple of questions that were in the chat. So I'll start us off with a couple of questions in the chat, and then if you have a question, feel free to unmute yourself and ask away. All right, so the first question is from Randy Bartlett, and Randy asks, any specific types of trees most used for roosting, species or size preferences?
Any specific, sorry, tree preferences? Is that, what was the question?
Yes. Sorry. Yeah, any specific types of trees most used for roosting, species or size preferences?
Okay. We are still ongoing with some of the evaluation of that. I don't think we can say specifically yet if there's any particular type of tree or species or size preference at this point. We have found them in a variety of different trees, and we're looking more at those tree metrics to see if there's anything that we can assess out of that.
All right, next question. Linda asks, any info on nutrient value of different prey taken by the bat?
I do not know the answer to that question. If there's different nutrient value and different prey taken, we will be looking at if there's any types that are selected more than what's available on the landscape, which might indicate some level of preference. But at this point, we haven't looked at nutrient value of the prey items themselves. Good question.
Phil Hester asks, Are mainland immigration still occurring?
That's a good question as well. Are mainland Hawaiian hoary bats still coming over to the islands here? Are mainland hoary bats still coming to the Hawaiian Islands? We are looking at genetic work right now to sort of suss out more of that. I guess the full history is still very much being sorted out and in the process of genetic work. So hopefully we will be able to determine a little bit more in terms of timing and how recently bats came to Hawaii, and if there are any still that are making it here.
Sierra and Emily ask, can we put bat boxes on our house for the hoary bats to roost in?
It's a good question, Sierra and Emily. Because these bats are foliage roosting bats, they will not use bat boxes. They've not been found to use bat boxes. Bat boxes are typically for bats that roost in crevices or cavities of trees, so that roost either under the bark or in a hollow section of a tree. And those are the bats that tend to roost in houses or churches, those kinds of things. That present sort of a wider space that the bats roost in. Our bats, because they just roost in the leaves up under the leaf clusters, have not been shown to use bat boxes. And not a lot of work has been done to look at what types of artificial roosts of any kind, if they would use any type of artificial roost structure.
All right, next question. Marcy asks, what type of thermal imaging units did you use for bat detection? Do you think a drone with a thermal sensor would be useful for roosting bat detection?
That's also an interesting question, Marcy. So the thermal imaging units that we use, we've used FLIR and Fluke hand thermal imaging units. I don't have any experience with drone thermal sensors. It would potentially be interesting to see. The main issue that we find with our thermal imaging is when the bat is blocked by leaves or other structures in the tree. So any kind of drone activity, it would potentially be challenging to get the right angle on the bat and not be blocked by the other tree structures. But that's interesting to think about.
So I have a question. So what would so I live on Oahu, so I was wondering if you have any ideas of where they would be on Oahu, or if not, where would the best place it'd be for public to see these bats?
I'm sorry, your video and audio was breaking up a little bit. Could you say that again?
So I live on Oahu, and so I was wondering where would the best place to see them on Oahu be, and I guess on the Big Island, or just in general, where would the best place for the public to see the bats? Is that?
I think you're asking, are there locations on Oahu to be able to see the bats? Is that what you're asking?
Yeah. If you know.
We have done some monitoring. We've done some monitoring work on Oahu, and there are bats there. Specific locations, I haven't done any of the work on Oahu, so I don't know those specific spots. Okay. But definitely at dusk is the best time to see them flying. And yeah, in areas where you can see sort of an open, if there's a forested area where you can see sort of an open clearing, that makes it a little bit easier to see them. Yeah. Maybe try that.
All right, we have more questions in the chat. Layla asks, Could you please elaborate more about Ōpeʻapeʻa general reproduction? It was breaking up when you were sharing.
Sorry, the video's breaking up again. Is this the question from the question about reproductive condition?
Yeah, so Layla Dudley's
Okay.
question about general reproduction.
Yeah, so in general, Ōpeʻapeʻa seem to have 2 pups annually. We don't know at this point if both of those pups make it to adulthood or not. They tend to breed. It seems like the males are reproductive in the fall, and they mate with females in the fall, and then the females become pregnant in the spring, and they're pregnant from the spring through the summer months, and the pups are born in the summer and stay with the mom until the early fall.
All right. The next question is from Mark. Is there any evidence to suggest hoary bats may be acting as vectors for rod via consumption of beetles and/or contact with beetle frass, which is then transferred to wounded ohia?
Thanks, Mark. That's a good question. We haven't looked at bats in relation to rod. That hasn't been part of our studies thus far. So I don't know whether or not that would be a concern or not.
So the next question is from Lee. Do the resources provided on this slide provide guidance for landscape architects' design notes and specifications provided to contractors whose work may affect existing tree stands, which may offer roosting habitat? Also, in places where barbed wire is required, is there a least problematic type known?
Thank you, Lee, for that question. I don't have the specific information for landscape architects or in terms of barbed wire. If you go to that website, though, you can find my email address and other folks on the project's emails addresses. And if you contact us directly, we could send some resources regarding those questions.
A new question from Kama. Why do bats get stuck/tangled on the top strands of barbed wire fences?
That's a good question. I do not know. I think there could be some potential to do some video monitoring of those, but it would probably be only in areas where it wouldn't be very beneficial on a wide scale. You'd have to try and be more specific with your sampling of that, which could be pretty difficult. So yeah, it's not clear. They definitely fly low in the habitat. They fly through trees and over grassy areas. So those fences are within the flight path for them, but I don't believe it's known yet why they become entangled on them.
All right, so Layla has another question. Layla asks, is there any association between Ōpeʻapeʻa and large bodies of water? I've observed them at dusk by reservoirs and also on the ocean near the coast.
It's a good question, Layla. I've also seen them in some places along the coast and over bodies of water. It's unknown if that they are particularly attracted to any of those particular locations, or if we can just see them better because it's more open, it's more clear. We're not having to compete with vegetation in the background that would potentially block us seeing the bats in the area. It also could be related to the insects that they're foraging on. So hopefully some of our current work will answer some questions about what types of insects they eat on the landscape and what they consume, and maybe help answer your question as well about whether or not those insects are associated with particular bodies of water or not.
From the chat, we have a comment from Randy. On Oahu, you could try looking around golf courses or trails like the Aiea Loop Trail or St. Louis Heights or similar locations. On Maui, I've seen bat activity on or near the former Kapalua Village course among the cook pine trees near the reservoir water hazard.
Great, thanks, Randy. I'm glad you're keeping an eye out for the bats. That's exciting. And definitely, yeah, good suggestions for places to see them flying at dusk.
We still have a couple minutes if anybody wants to unmute and ask a question.
I got one more question. So, are there more prevalent, like, are they more common in lowland areas or in like higher elevations?
Thanks, Nathan. That's a great question. We are still trying to assess that with our current work. It seems like there might be some seasonality to where they are located in lowlands versus high elevations. We have been able to capture bats over the past 2 years at low elevation and high elevation throughout the year. So yeah, it's definitely something that we're still trying to assess a little bit more. But good question.
We have another question in the chat. Linda asks, how long do the juveniles stay with the mom?
So the juvenile bats we're monitoring pretty actively right now with video footage, and we'll have more specifics for Hawaiian Hoary Bats coming down the road, hopefully. In general, bats of this size and this group tend to stay with the mom for the first 2 months or so after birth.
Bob O has a comment. Bob says, I've seen them at shoreline up to tree line on MK. So I assume MK is Mauna Kea.
Yeah, we do net sort of all along that area, and we definitely have seen them throughout the whole landscape. So they seem to, they're difficult to catch and difficult to follow, but they are somewhat widely distributed. We're not sure about the population numbers still, but they seem to cover quite a bit of the landscape.
All right. Elliot asks, is funding secured for long-term population monitoring, e.g. data collection to come up with population estimates and determine long-term trends in abundance, etc.? Thanks.
The project that we're currently working on is funded through 2021. And beyond that, I don't know about our future efforts and work. And in terms of population monitoring, yeah, it's definitely something that requires a large data set and continued work at pretty high level of effort. So with our efforts, we'll certainly try and provide as much information as we can for those estimates, and we'll see where it goes.
So at this point, we're running a little bit over. As long as Julia is free, we can continue to answer questions, but we understand if some of you have to run and go back to work or whatever is happening in your life right now. And know that if you do have to leave right now, but you're interested in these questions, the video recording of this webinar will be uploaded to YouTube and will be added to a playlist that we link to you guys in an email with a feedback form and some other information after the presentation ends. But yeah, so I will go through what else is in the chat.
Right. Yeah, thank you all for attending. I really appreciate it, and for all the questions. It's great to interact with folks and explore, explain some of the cool things about the bat that we have here. So thank you all.
All right, so Bonnie says, Nathan, look near reservoirs. Once you recognize flight pattern, you will be able to see in other spots. I've seen driving home in my Hilo driveway. Layla asks, how long is gestation period?
Sorry, your video broke up. Layla, question about how long is gestation period?
Yep, that's the whole question.
Okay. We don't have specific timing for that. From other species, we believe that the bat stores sperm through the winter months and then becomes pregnant in the summer. In terms of observable time period, we tend to see pregnant bats in May and June, and then we see lactating bats starting sort of end of June into early July. And so that time period is sort of the period that we know of from our bats at this point.
Awesome. There are lots of people in the chat thanking you and telling you that this was an excellent presentation. So I figured you should know. There is another question from Sue. When you take a tissue sample, where is the sample taken from?
So when we take tissue samples, we take them from the wing membrane. We use a small dermal punch, a 3mm dermal punch. This is pretty common practice with bats. The wing tissue is a very fine dermal layer, and it heals within a few weeks to a month. And so if the bat's wing was outstretched, we take sort of a small hole, like a ear punch through, like a earring punch through their wing membrane.
I think that is all of the questions that we have in the chat. So if anybody has any final questions or comments, please unmute yourself and ask them now or put them in the chat, but otherwise we are probably going to end in a few minutes. So with that, I think our presentation is over. Thank you all so much for attending. We really appreciate it. And it's so great to see how enthusiastic you guys. We had so many wonderful questions. So keep a lookout for an email that will have our feedback form. Please fill it out. We will also have in there the link to the presentation for October. That will be Ian Ware presenting on what HipNet is currently up to. And it will have the link for the current YouTube playlist. This recording should be in there by the end of the week. So yeah, thank you so much for the amazing presentation, Julia. That was really informative.
Thank you, everyone. Appreciate it.